Okay, so good evening, everybody. I think Victoria just joined us. How are you feeling? I thought you were sick yesterday, right? Better, that's great. All right, so last week we talked about the drug development. We started talking about the drug development process, and we focused more on commercialization last week. So how do we position the product ahead of approval in terms of marketing, how we are going to find the right markets, the right patients, etc. Okay, so today we start talking about the drug development process per se, and in that whole process, the first step is, of course, drug discovery and then preclinical studies. So that's what we are going to focus on in today's lecture. Okay. Gerardo, how are you? Jerry, sorry. Okay. So as I had mentioned in the course materials for today's lecture, you might want to read the textbook chapters 2.1, sections 2.1 through 2.4, and 5.1 through 5.5. So this section of the book talks about the discovery research, and this section talks about preclinical studies. Okay, and here are a couple of other resources for you to view on your own. I think I have put the pharma brochure online, but you can always go there. And also this website, if you go there, there are some cool videos talking about the drug discovery process. So you might want to check that out on your own. Okay. So, a definition of a drug is an active ingredient that is intended to furnish some kind of pharmacological activity or other direct effect on any of these things: diagnosis, cure, mitigation, or treatment. So it doesn't have to be a cure. It can mitigate. So for example, if I have a cold and I have a headache, an aspirin will mitigate my headache, right? It doesn't cure it literally. Even treatment of some kind or prevention, so vaccines are included in that, of a disease, or affect the structure of any function of the human body, but does not include the intermediates used in the synthesis of the ingredients. So for example, if you are making aspirin, and part of making the aspirin is another intermediate ingredient. So you start out with one chemical, and then you convert it to another, and then you convert it to the aspirin. Then aspirin is the one that has the activity, and that's the drug. This intermediate is not a drug. Okay, so that's the basic definition of a drug. So last week we talked about the timeline, of course, this famous 15 to 16-year timeline, and here are the human clinical trials. We will be talking about those later on, and today we are going to focus on this portion of it: discovery and preclinical. There are various abbreviations here. I think everybody knows FDA, Food and Drug Administration. That's the agency that we apply to get marketing approval. Not only marketing approval comes over here, but we also apply to them to get approval to even begin clinical trials, and that comes right here. And so we have to go to the FDA at each of these stages with more and more data. Okay. So, but before you go to the FDA, you want to, of course, find a drug and do some preliminary studies with that drug to to go take the data to the FDA. So that's what we are going to talk about today. And then IND stands for Investigational New Drug Application. So IND is filed over here after you get preclinical data. BLA and NDA, they are the same things, but they apply to different products. So for example, BLA is applicable to biologics, and mostly this covers the biotech products, right? Biologics. So anything that is derived from biological sources. So most of the proteins that are derived from biological sources are covered by BLA. So if you are developing a biotech product, you are more than likely to be using a BLA rather than an NDA. NDA stands for New Drug Application, and that, of course, is mostly used for smaller molecules. Okay. So it used to be that the biologics application went to this BLA, CBER, Center for Biologics Evaluation and Research, and the NDAs went to CDER, Center for Drug Evaluation and Research. But this boundary has been shattered now. So a lot of biologics now mostly go to CDER. Okay, so the BLAs are also going to CDER now. There have been many changes in the FDA lately. Okay, so we talked about this before. The timeline is 10 to 15 years, and the average cost is about $1 billion, includes the cost of failure, and you get one approval for every 5 to 10,000 compounds that enter the pipeline. Very, very slim odds. Now this, of course, applies mostly to small molecule drugs. Okay. Now, small molecule drugs, because they are not very specific, proteins, when we talk about biotech products, we are talking about proteins. The action of proteins is a lot more specific. So the chances of success are a lot higher with biotech products than what we talk about here, okay, which applies to small molecules. Okay, so essentially these are the odds that we just talked about, and so here is the number of compounds that you are testing at each stage and the typical cost. Okay, so in discovery research you might be testing 5 to 10,000 molecules and spend about $300 million on it. And then once you have identified out of those 5 to 10,000, 250 molecules, and those are entering preclinical testing, which means it can be either lab testing or animal testing. A significant amount of dollars spent there, and then only 5 to 10 out of those might go into phase 1 clinical trials. And as you go forward, the approval, you might get one approval, after testing all of these compounds. So significant risk and significant outlay of capital is involved. So it behooves us to actually, so this is a picture from that pharma brochure that I talked about, that I have given you a reference for. It's showing the same thing more visually, starting with 5,000. Here is your drug discovery and preclinical, and then focusing more on the clinical trials and approval. There. Same idea. Okay, so when you want to discover a new drug, there are many, many hurdles to discovery of the new drug, right? So a good way of understanding what those hurdles are is to think about how a drug candidate can fail. And there are so many ways a drug candidate can fail, right? You might have a really good molecule, but when you take it when you take it orally, it may not dissolve. right? It may not dissolve in your stomach. It may just pass right through. And release, is it fast? So dissolution and release, is it fast or slow? You want it to dissolve really quickly, and you want it to be stable in your stomach, right? Now, so interaction with digestive enzymes. So this is one of the main reasons why proteins are not taken orally, right? Proteins are easily digested in the gut. So you might have a really nice protein and you take it orally, and it will break down into its component amino acids, and your body might make different proteins from it. So most of the proteins are injected. So proteins are mostly injected. So this really applies to small molecules, right? So small molecules will pass through the barrier from the stomach line, from the intestines into the bloodstream, but some of it might be digested. So how much of it is available, how stable it is. So this is sometimes called bioavailability, right? So many times people will study bioavailability, how much of the drug can actually pass through. So if it is just 1%, so think about the implications on manufacturing. If your bioavailability is 1% compared to 10%, well, if the bioavailability is 1%, you're going to have to manufacture 10 times as much drug as if the bioavailability were 10%. Does that make sense? So it has major implications on manufacturing. Also, can it pass through the walls of the small intestines? Is it absorbed? Okay, so that's another. So stability and absorption, two different things, right? Can it pass through? And then once it makes its way into the bloodstream, how is it binding to the receptors that we are thinking about? The receptors that are implicated in the disease state, right? Or sometimes the disease is being modulated by some kind of an enzyme or other target, right? So how is the binding of the molecule that you're testing to these different things? This is to see how effective the drug is, right? Now sometimes the drug might be effective, but it also binds to other undesired targets, and in that case it will create side effects. So it may be effective, but it may have too many side effects. So you know the story about aspirin, for example. Aspirin is very effective in treating pain, but it also has side effects on your stomach. Your stomach lining, for example, it will hurt you. You'll have problems with digestion, and also it can lead to bleeding, right? So those are some of the side effects. So instead of that, some of the newer products like Celebrex do have a good. They are effective in treating pain, but have less side effects in terms of stomach problems, right? So that's what we mean by binding to undesired targets. And then finally, how fast is the drug broken down by your body? Obviously, at some point, the drug is going to be broken down and eliminated, excreted from the body. Whether it is fast or slow, that's going to determine frequency of dosage, right? If it is eliminated pretty quickly, then you will need to give the drug more often, and that's inconvenient sometimes. So That is also an important aspect. So when you are looking for a good drug, you have to consider all these aspects. How can the drug fail? And so, there are many times you will have many drug candidates and they will fail in one aspect or the other. Okay, so let's now talk about the two aspects. The first one is discovering a new drug, and then we'll talk about preclinical studies, right? So how do you go about trying to find a new drug? Well, first of all, you have to identify unmet medical need. Is there a medical need? If you have lots of, for example, you might ask the question, I want the drug to lower cholesterol. Well, the question is there a medical need? Yeah, there is a medical need, but what's the market potential? Well, there are already some drugs on the market and they seem to be very effective. So what's the idea behind coming up with another drug to treat the same target, right? On the other hand, you might come up with a medical need where there are no other treatments, and that will be a good target to use. What's the prevalence of the disease? You might come up with a medical need. For example, I don't know if I talked about the drug leptin, but it's not a drug. It's not a drug, but it's a hormone, leptin, that affects fat cells, right? So this affects fat cells, and so it turns out that there may be like 10 or 15 people in this world, in this whole world, that have a deficiency in leptin, and they are obese. They are morbidly obese, and for them, if you give them leptin, they will lose weight very, very quickly. But having a patient population of 10 to 15 people is not going to be very attractive in terms of market potential, right? So remember, Amgen paid $76 million to Rockefeller University for the rights to this drug, and they found out later on that there are 10 to 15 patients that fall into that category. And that's because initially they thought that they may be able to use leptin on the general population, just like we use insulin on a lot of diabetic patients, right? But it did not turn out to be that way. So prevalence of the disease and market potential are two important questions to answer when you are looking for a new drug. Do we have a biochemical target? So for example, you might have symptoms. You might have a disease that you know the symptoms for, but you really don't know the mechanism of the disease. And if you don't know the mechanism of the disease, it's hard to come up with a drug, right? You want to know what to interfere with, what in the body you want to interfere with, right? So you should have a clear biochemical target. And then, are you able to synthesize the compounds that are selective for that target and are bioavailable? So those are important questions as well. And are the compounds efficacious in disease models, show dose response, and are not toxic? So these are all important questions to answer when you are discovering a new drug. Okay, so today's emphasis, as I mentioned, is in these two areas. And while you are doing drug discovery, manufacturing is also affected. How is that? Because when people are testing new molecules, you need to be able to produce them. So people in process development and R&D manufacturing. There are groups there are groups known as process development and R&D manufacturing. Sometimes clinical manufacturing is combined with that. So these groups get involved in trying to figure out a process to produce these products. Unless you can produce them efficiently, then it's not going to be very useful. So manufacturability, can you manufacture the compound easily, has to be considered when you are choosing a molecule. Sometimes you have two candidates. One may be slightly more effective than the other, but the second one is more manufacturable. Then you might want to go with the second one, even though it's slightly less effective. Okay, so when you are discovering a new product, many disciplines are involved. Usually there is a product discovery team, and it includes, of course, pharmacology, pharmacokinetics. There is a project management, project leader involved, and so the project management group gets involved. There is, of course, we're going to talk about pharmacokinetics and pharmacodynamics today. Molecular biology, process development, molecular biology to come up with the DNA sequence. Regulatory is, of course, involved, looking at what data needs to be generated to give to the FDA. If it's a small molecule, chemistry is involved, toxicology, biostatistics is involved to figure out the data. Combinatorial chemistry, of course, if you are doing small molecules, they are coming up with lots and lots of different compounds to test. And high throughput screening, when you have lots of different compounds, you want to quickly test it, and that's called high throughput screening. So there are many disciplines that are involved in the discovery, even in the discovery stage. Okay, so these are the 4 steps for discovery. First, you want to identify a target in the body. You want to identify a target in the body for a disease state. Okay, and once you have identified the target, you want to validate it. You want to make sure it is the correct target. So target identification and validation. Once you have identified the target, you will start testing compounds and identify a lead compound. So that's your first compound that you think is going to have interesting activity in treating that disease target. Okay, and so you identify lead compounds, but then you want to make certain changes to the leads to see if you can improve the properties of that lead compound. So that's called optimization. So you might want to play with the structure of that compound a little bit to optimize the properties. And then comes preclinical development, where you are testing it in the laboratory, certain lab assays, biochemical assays, as well as in animal models. So preclinical development involves laboratory assays as well as animal testing. And at that point, you are ready to make a decision to go into humans for phase 1 trials. So this is called first in human or first in man decision. Okay, so today's we're going to talk about, before we go into clinical trials, what are the steps. Okay, so let's say you have some kind of a target. Companies can take 2 different approaches, either the irrational approach or the rational approach. Now, irrational approach is kind of the old method, where people would just try out different chemicals that are in their storage, or they have found these chemicals from maybe plant sources or other sources, and they just take a hit or miss approach. So it's called irrational because you don't really think about the target. You're just testing lots of different chemicals. Okay. So basically, you relies on screening of many compounds in the hopes of finding a hit. Okay. So that's irrational, and it used to be the more common approach because we didn't have a lot of the sophisticated techniques we have now for the rational approach. Now we do have lots of techniques for the rational approach. So the book, if you read the book, it gives you an analogy of lock and key. This is akin to finding the right key for a particular lock. So the lock is what we call target, is the target, right? So you have a target in the body. You need to find the right key for it. And so the key is your lead compound. Okay? So an irrational approach is akin to just trying 1,000 keys to see if any of those keys fits the lock. Right? That's kind of a hit and miss approach. You just collect a bunch of keys and just keep trying whether it fits the lock. Okay? A rational approach would be to try to find the structure of the lock, internal structure of the lock, and then designing a key that will fit it. Now it will take a little while to figure out the structure of the lock. But once you know the structure, it's probably easier for you to find the right key. Yeah. So that's the rational approach. How are we doing? I don't hear any questions. I just want to know if everybody is understanding what I'm saying, if there are any questions. Okay, good. Okay. So let's talk about target identification. So what I've done here is to just give you an example of how a whole team of experts might identify a target. So first, a team of different types of scientists, molecular biologists, cell biologists, biochemists, and they are in collaboration with a teaching hospital. They search for a novel target protein involved in inflammation. So the target area is inflammation, but they are kind of looking for a protein that might be involved in the whole pathway of inflammation. Inflammation is a complex pathway, and it has many, many different molecules and intermediates involved. So you might be targeting any one of those proteins. Okay, so they try to identify a new target protein in the inflammation pathway. Okay, and the team then forms a hypothesis about the specific pathway. They hypothesize that this protein is key in modulating inflammation response. So inflammation can happen in the body in response to many things, right? If you get a cut, you get an inflammation. If you have some kind of infection, you might get inflammation. Many different things will trigger inflammation pathways. So you want to target some particular protein there. Once you have a hypothesis, the molecular geneticists will identify the genes of interest for that particular protein. So that's how molecular genetics, molecular biologists get involved. Then you might use a technique like DNA microarray technique. DNA microarray is a technique where you have a gene, literally a chip made of some kind of a glass that has lots of tiny wells. So it's almost like a silicon chip, right? But it has tiny wells, thousands of wells, and in each well there is a different gene. So you have thousands of genes that are spotted onto these wells. Okay, And what you do is you take a sample from a particular tissue or affected area. You want to know which genes are active or not active in that uh, affected area. So you are going to use DNA, and this is specially. So I'm not going to go into a lot of detail here, but you find something. You make something called cDNA from a tissue sample. cDNA is the DNA that is being expressed in a particular area, and you label it so you can identify it. You label it with a fluorescent label or a radioactive label, and then you put your sample on this gene chip. And what happens is the genes that are being expressed will light up. So you will have certain wells on the gene chip light up, and that tells you which genes are involved in that disease state. Does that make sense? So you could get samples from a deceased person, I mean not a disease, a person with a disease and a person that is healthy, and try to identify what are the differences in the genes being expressed this way. Okay, so that's called DNA microarray technique. And so you can get data on thousands of genes and find out which genes are involved. And then there may be a group in your company called the bioinformatics group, and that basically deals with lots and lots of gene sequence data that may be available either in a public library or their own proprietary sources. So they may look through the reams. Nowadays there are so many genomes that have been sequenced, so there is lots and lots of genome data available, gene sequence data. So you might look through all the data, the bioinformatics group, to find potential gene sequences. So this way a set of potential targets is identified, okay, by this team. And then what you do is you continue. The protein chemists now get involved, and so they knowing the sequence of the gene, they can synthesize the target proteins, and they analyze it to make sure that they can be appropriate drugs. Remember we talked about how drugs can fail. So they look at the properties of the protein to make sure that this protein may be appropriate. And then they will analyze the structure of the protein to see if there are appropriate binding sites. And that will rank the potential targets. Knowing the structure of the protein, it tells you whether you have a good target or not, right, for a potential drug. And then further analysis might indicate that one target is structurally similar to others that have been targeted by oral drugs. So that gives you a good idea that this may work, right? And so you can then validate the target. Remember, we have a target now. They have identified a target, but you have to validate it. So there is a gene involved, right? There is one particular gene involved. So what they do is they take a mouse and they either remove the gene or make that gene ineffective in that mouse. So they literally knock out the gene, that particular gene that you are interested in, from the mouse. And so that's called a knockout mouse. So you have literally knocked out a particular gene from the mouse. This is very useful because now you can see if the mouse is exhibiting the disease state. That makes sense? So that validates your target. By using a knockout mouse, you can validate the target. Also, you can use the knockout mouse to try your treatments, right? You can give the molecule that you're figuring to use as a drug to this mouse and see whether it works. So this is how we can validate a target. Now, knockout mice are one way to validate a target. Of course, the people use different ways to validate targets. They might use some kind of a biochemical assay or a cellular assay to validate the target. Okay, so this is just one way, but it's becoming very common, especially when you are using proteins as the targets. Any questions so far? So right now we talked about target identification. Target is the part of the body that you want to affect, right? Where you have a disease going on. So the way you affect it is by identifying a compound, right? And so we call the compound that you identify that will be used as a drug as the lead compound. The main compound that you identify is the lead compound, okay? And so you identify, once you identify the target and validated it, now you want to identify different molecules that will have some kind of activity or affect the target in some way, right? So it should bind to the target, or it should turn off the activity of the target, or it should enhance the activity of the target, whichever the case it might be. Okay, so you're going to identify a lead compound, and then you're going to optimize its properties. Okay, so the following is an example of a small molecule lead compound. So once you have identified a target, discovery chemists will generate hypotheses about how some molecules may interact with the target. So for example, if you know the target is an enzyme, so it might be a protein. Enzyme is a protein, right? And it has a certain three-dimensional structure. Proteins have three-dimensional structure. So they say, oh, this is an interesting area right here. And if I construct a compound that has this particular shape, right, then it might actually fit right there. And so, so they will say, oh, I can construct a compound that fits that molecule, okay? And it will, and because that particular, if it fits that enzyme in that place, it might inhibit the activity of the enzyme, okay? So the chemists, based on their knowledge of the structure of the target, can propose molecules that might be able to bind to that compound. Okay, so then the computational chemists use computer modeling to identify molecular structures that will bind to the target. Okay, and then medicinal chemists synthesize the compound, synthesize the candidate compound, and then so you have lots of different compounds that have been synthesized, and so the high throughput screening group, HTS group, gets involved and tests all the compound on the target protein. Okay, so after you test it, what you're doing is you're looking for certain activity, certain binding activity, and by doing that, you are identifying hits. So these are among your lead compound. These are your lead compounds that you're identifying. Okay, and so once you have identified some hits, then you can also look for analogs of that molecule. That means slightly different structures but similar compounds, or derivatives of the lead molecule that may have better properties. So what you're doing is you're optimizing the lead, right? And so after optimization, you have identified a candidate molecule that has the best fit to the target that you generate. So this is called lead identification and optimization. Now, the example I have given you is for a small molecule, right? But you can think of similar ways people might identify a lead compound for a protein molecule. So but different people will, different types of scientists will be involved then. For here we have chemists. Over in the protein side, you might have protein chemists, you might have molecular biologists involved. Okay. All right, so where do these leads come from? Okay, so leads come from a variety of different sources. So you might be randomly screening existing, most companies will have a library of compounds, small molecule compounds that they routinely test. So random screening of existing or newly synthesized small molecule compounds. There are combinatorial chemistry libraries. So these are compounds that are slightly different from each other, but a whole slew of compounds that you can try. You could isolate compounds from natural sources like plant extracts and things like that. Or, for large molecules, you might have proteins from, from the body or from animals, etc. Okay. You may also get leads from universities or other companies, so you can license in lead compounds from other companies or other universities. So there are many, many different sources where the leads can come from. Okay, so once you have identified a lead compound, when a single development molecule is identified, the molecule now moves out of research, and now you are starting to think. Now you're starting to think about clinical trials, phase 1, first in human. But before you do that, of course, you have to do many other things. So far we have talked about discovery, right? So you have discovered a new product, a new lead compound. So we have talked about discovery, and before you go to clinical trials, we need to talk about preclinical studies. Okay, so what are the preclinical studies? Okay, so preclinical studies are performed typically with one or more, typically with 1 to 5 optimized compounds. Okay, because you are not yet sure, you don't have the exact one. You might try a few in preclinical studies to focus on one of them, right? So we need to do extensive laboratory and animal testing to determine if the candidate molecule is safe enough for human testing. And also, you want to know how the drug works and what its safety profile looks like. Okay. So at this point, you also have the opportunity to address manufacturing, because now you're going to need larger and larger quantities of the material. So you are going to start to scale up your manufacturing. And a lot of times, problems come up at this stage, and you need to solve them right away, right? So there are many tools available. You want to find the right approach. Sometimes you want to try to use a mechanistic approach. So you want to make sure that you know the mechanism of how the molecule is acting in vivo in animals. Sometimes you don't know the mechanism and you use empirical approaches just to see what the effect is, right? So you're not doing detailed testing, but just looking at the broad effects. Now, to find the right compound, you want to make sure that you have some kind of an efficacy model, whether it is in vivo, that means in animals, or you might be testing it in cells, just tissue culture, right? You have to have the right enzyme or receptor assays. These are the binding assays that we talked about. And you need pharmacokinetic and pharmacodynamic models. We're going to talk about what PK and PD means in a minute. You want to make sure that it's not toxic, and you could use computational techniques. Sometimes nowadays people are actually, and you might, if you have read the sections of the book I talked about, he talks about in silico testing. What does that mean, in silico? So there is in vitro testing, there is in vivo testing, and there is in silico testing nowadays. So, a few years back, you would only talk about in vitro testing and in vivo testing, right? In vitro means you are testing it in the lab with some biochemical assays or cellular assays, right? In vitro means in glass, in test tubes, right? But in vivo means you're testing it in real life. So you're testing it in animals. In vivo means in life. In vitro means in glass. So this is in lab testing. This is in animals. So what is in silico? In silico means in computers, right? So this is in, so you're basically searching databases. Nowadays, there are so many databases that you can actually run simulations in computers to see if your molecule is going to have an effect. Does that make sense? So these are the different tools that are available for testing. Okay, so we talked about pharmacokinetics and pharmacodynamics. What's the difference? Pharmacokinetics describes what happens to the drug in the body. In other words, what does the body do to the drug? Okay, pharmacokinetics. What does the body do to the drug? Does it break it down? How long is it going to last in the body? Okay, so here we are looking at how the body treats the drug. Okay, you want it to be stable. You want it to be, so for example, when you put the drug in the body and you look at the concentration of the drug in the blood, initially you might get a real spike. And then over time, the body is going to break it down and clear it out of the body. So this is PK, looking at what the body is doing to the drug, right? Now PD, on the other hand, looks at what does the drug do to the target. Okay, what does the drug do to the target? That means, does it have an effect? So you might be looking at, if you're testing a painkiller, you might be looking at, complaints about pain. Okay, so you're asking the patient, does it hurt now? Does it hurt now? And the patient is reporting, oh no, the headache is gone. So in a few minutes, the patient will report that headache is gone. So you have a good effect, right? And over time, after some time, as the drug wears off, the patient starts to complain more and more about the pain again. Oh, the headache is back. So over here, the headache is back. So this testing of the effect of the drug on the body is pharmacodynamics. So is this clear, the difference between PK and PD, pharmacokinetics and pharmacodynamics? Pharmacokinetics is a look at what's happening to the drug in the body over time. And pharmacodynamics is looking at what effect the drug is having on the body over time. Okay, both are important. What if the drug clears out of the body really quickly within a few minutes? Then it's not going to be useful, even though it has a really good effect. So PK and PD are both important, right? So PK/PD, the Department of Pharmacokinetics, Pharmacodynamics links PK and PD so that the time course effect is described for a given dose regimen, right? So for a given dose regimen, you want to know how the drug is affecting the body and how the body is affecting the drug. Show of hands if you're still awake, getting something out of the lecture. Anybody? Okay, I see one hand. All right. Thanks, Jerry. Let me know if you are here, you are understanding the lecture, if you have questions. Okay, I need to see something happening from the audience, otherwise I feel kind of lonely. Jonathan, Pat. Okay, Victoria is there. I see that. Okay. Tim, great. Okay, I feel better now. Okay. All right, so great. Thank you. All right, so now we talked about PK/PD. So what do we typically look for in PK and PD? So for example, here is a graph of concentration of the drug versus time. This might be the concentration of the drug in the bloodstream over time. And you can see that the drug concentration is going up over time, and it has leveled off in this range right here. And so this is the pharmacokinetic aspect. Now, the pharmacodynamic aspect is that the drug is toxic beyond this level. Okay, you don't want the drug concentration to go beyond higher than this, and the drug is not effective below this level. Okay, so this is the subtherapeutic level, and this is the toxic level. So the target for us is to make sure that the drug concentration in the blood, in the plasma, remains in this range, right? So the dosing has to be such that you are trying to target a concentration right in the therapeutic range. So what this graph is showing you is that there is a therapeutic range, which is this range right here. This is the therapeutic range. Beyond this is the toxic range, and below that is subtherapeutic range. So this is how, by doing testing, PK/PD testing, you are actually finding out what kind of dose regimen to give, right? To be in the right therapeutic range. So this is called the therapeutic window right here. This is the therapeutic window for the drug. Okay. Now, when you give a person a drug, many things can happen to it. You want to study many things. First of all, you want to know whether the drug is absorbed. Okay, does it go through the barrier? So, for example, if you're taking an oral drug, does it go through the stomach lining into the bloodstream, right? Through the intestinal lining, to the bloodstream. How much of it is passing through, and how much of it is not passing through? Bioavailability. Sometimes if you are targeting something in the brain, does it cross the blood-brain barrier, right? So that's absorption. Then, once it has gone into the bloodstream, how is it distributed? Is it concentrated in certain tissues, or is it going through the whole body? Okay, so how does it distribute between the various tissues and at what rate? So distribution is important, right? You want the drug to go to the place that you want it to go to, not undesirable places, right? So that's distribution. And then metabolism. Is the drug changed by the body? Do certain enzymes in the body break it down? Especially, we know that the liver will often break down drugs. So metabolism. So metabolizing enzymes, what effect do they have on the drug? And finally, how is it eliminated? How fast is it eliminated, excreted from the body? In what form and by what routes? So these studies, these are all PK studies, and they are called ADME: absorption, distribution, metabolism, and elimination. Okay, ADME studies are PK pharmacokinetic studies. That's what we are studying when we do ADME, when we do PK. Okay, so PK/PD. Then we also need to look at toxicology, right? Toxicology is the drug toxic in an in vivo setting? So sometimes, there's always concern about using animals because people don't, many people have objections to use of animals. So companies always try to look for cellular models first. You can treat certain tissue cultures with the drug to see if it is toxic to the cells. And you might want to use particular types of cells to see if they are toxic to those particular types of cells. So first you try to use cellular models, and then you might want to use animal models. And by using animal models, what you are trying to find out is what is the maximum tolerated dose and what is the minimum effective dose. Remember, that's the therapeutic window. So beyond the maximum tolerated dose, it's toxic, and below minimum effective dose, it's subtherapeutic. So this is the therapeutic window. Okay, and then you're doing many different safety studies. They are defined by ICH. ICH is International Conference on Harmonization. Okay, ICH stands for International Conference on Harmonization. Basically, scientists from many different countries get together and they figure out what are some of the standard things that they are going to do when they do toxicology testing. Okay, so single, multi-dose, rodent, non-rodent, whether it is carcinogenic, is there any reproductive toxicology, what kind of kinetics you observe over time, and if there is any immunotoxicology, is there any immune response, things like that. So these are all toxicology studies that need to be conducted in cellular models first and then in animal models. Okay, so when you figure out some animal model, you want to make sure that it is predictive. Okay, if I use a certain mouse and I look for toxicity in that mouse, is this going to be the same toxicity profile that I get in humans later on? So, sometimes people use the wrong animal model, and that many drug candidates fail because insufficient animal models are used in preclinical testing. So the people in people in toxicology, you need to have really good people who have studied this kind of thing. How predictive is a particular animal model in humans, right? So does it have useful actions in preclinical models? Does it have any unexpected actions? So you want to study that. You want to choose the right animal models and the right number of animal studies to be confident that when you move into humans, you're going to have good results. So preclinical studies before IND stage, what you want to find out during those studies is you want to identify the starting dose for human studies. You want to make your best guess as to what starting dose you're going to use in humans, right? And you want to make sure you understand what kind of toxicities to expect and whether you can reverse those toxicities, right? Whether you can stop dosage and it will be reversed, right? You don't want chronic toxicity. You don't want something that will affect the human body, permanently. And so you need to have some kind of a guide for dosing regimens and escalation schemes. That means normally you would start off the human studies at a certain dose, and over time you want to then try a second dose and then a third dose. So this is the escalation scheme. So you want to have that available before you start clinical trials. You're going to propose this to the FDA saying, these are the doses we want to try. This is the data. These are the data that we have collected from animal studies, and we expect these kinds of results. So you want to be really prepared when you go into human testing. So the study design, you want to make sure that you have the safety and start dose decision that you should be able to get out of the study design. You want to try to mimic the schedule, duration, formulation, route, etc., as much as possible to the clinical trial. So you can see how this works. You are visualizing, so you first visualize a clinical trial. What you're going to do in the clinic, right? Now, based on that, so you're saying, I want to try 3 milligram dose or 10 milligram dose of this drug in the humans. Now, what you do is, suppose you have your animal model is a mouse model. So then what you want to do is to, a mouse is usually a few grams, and humans are weigh kilograms, right? So you want to scale down the dose appropriately, so many milligram per kilogram. So you want to scale down the dose and then try the appropriate dose in the mouse to see what kind of effects you get. So you want to, that's how you mimic the schedule, duration, formulation, etc. Now you also know that in humans the drug might last, few hours, whereas in mice it might last less than that. So you want to modulate the duration, formulation, etc. accordingly. Okay. It should conform to the standard toxicology protocols that have been developed, and very important, you want to conduct the experiment according to GLP. GLPs, remember what GLPs are? You know what GMPs are. It's good manufacturing practices. GLPs are good laboratory practices. Okay. Very similar to GMPs. Basically, you want to make sure that you write down what you're going to do, and you document everything, and you do everything according to the protocols, right? And these GLPs have been encoded in 21 CFR 58, Part 58. So Code of Federal Regulations, number 20, Title 21, Part 58. They spell out the requirements for non-clinical laboratory studies intended to support INDs, NDAs, and BLAs. So we want to make sure that we follow the right protocols. And so some examples of GLP requirements. And in the book, they give you more examples of that. So these are just examples. Trained personnel, you want to make sure that you have hired people who have the right training. And if they don't have the right training, you have given them the training and trained them and documented the training. So you should have their job descriptions on file and their resumes on file, etc. You want to make sure that you have defined the protocols beforehand, and then you conduct the experiments according to that. You want to make sure that there is an independent quality assurance unit that is reviewing your protocols and reviewing your results. You have to have good systems, quality systems for data storage, deviations, and reporting. So oftentimes people will invest in something called LIMS, Laboratory Information Management System, right? So you should have a good LIMS system that can generate the right reports for you, that can store the data appropriately, etc. So these GLPs were first implemented in New Zealand and Denmark in the 70s. In the US, they were implemented in 79. And the international GLPs were adopted in 1981 by this OECD, Organization for Economic Cooperation and Development. Okay, so GLPs are really, really important in preclinical testing, and you might get inspected by the FDA for this. Okay, it's less likely than manufacturing plants. Manufacturing plants get inspected more often by the FDA, but it has happened. FDA has gone into laboratories to check their GLP status. And sometimes what happens is they will go into a company to inspect their manufacturing plants, and then they will say, oh, we would also like to see your animal laboratories to see if you're following the GLPs. Right? So it's important that GLPs be followed. All right, so preclinical tests help you to validate animals as predictive of human condition. So you have to have the right models. Establish safety margins between the therapeutic window. General safety in several species. So you might test it in different species of animals, mice, dogs, monkeys, etc. And understand the response as it relates to metabolism, how the drug is treated by the body. And you are able to then optimize the dose range, formulation, and frequency of administration. Okay, so that's what the PK/PD studies do for you. As I've been talking throughout the lecture, manufacturing also benefits from effective preclinical studies because we are slowly escalating the amount of product needed, right? From small studies to larger studies, etc. You need more and more product. So it allows development of a scalable process that is well characterized. You want to make sure that your manufacturing is reproducible, and you can do it. You can produce the same type of product again and again at a large scale. It provides data to determine manufacturing strategy. You might try out 2 or 3 different processes, and you might find that one process is better in terms of reproducibility than the other, so you would go with that. For example, at this stage, you might be trying out different hosts for if you're manufacturing a protein, you might be trying out different hosts for your protein. So you might be looking for E. coli versus CHO cells versus yeast cells. Okay, so you might find that, oh, E. coli produces a lot of protein, but it's not very active, whereas CHO produces less, but it is more active. So you make decisions about manufacturing at this stage. People are also developing the right formulations what kinds of excipients are you going to use um, with your drug products, what kinds of buffers you might use, things like that, the formulations, right? And of course, dose range. It allows for most efficient use of the compound. So, what we have talked about today is we talked about this portion, how you discover a product, looking for different targets, and then identifying lead compounds and optimizing the lead compounds. We also talked about validating the targets, and then what we do in preclinical development. How do you figure out, before you go into humans, what kind of doses you might want, how do you identify the toxicities, potential toxicities, things like that, right? So we talked about pharmacokinetics and pharmacodynamics, how the drug behaves in the body and how the drug affects the disease state, right? And then we talked about the therapeutic window, how you determine the therapeutic window, and toxicity in animals, first in cellular assays and then in animals. So we have come up to the point where we are ready. Hopefully your drug has worked well. In these, you have identified a drug and it has worked well in the animal studies, and now you are ready to go into first in man or first in human, and we are going to go into clinical development next, right? So we have come up to here, and it is a long road, as you know. So far, how much money have we spent? Anybody remember? You are developing a product. How much money have we spent so far? I believe in the screening of the compounds, it was around 325, if I remember right, million. And a few hundred, another, I forget now, 70 to 80 million in the preclinical testing, right? Well, the numbers are there in the table. Anyway, so we have spent a good chunk of money, and hopefully when we go into clinicals, we will get good results, right? But we know the odds. Okay, what questions do you have? Now, I noticed that most of you are participating in the discussion forums and also have submitted your quizzes and assignments. That's great. Those of you who still have not started doing that, you should do that quickly. Otherwise, you will be losing points on the grades, right? So I want to encourage everybody. So there are 3 things going on each week, right? So I want to make sure everybody understands the 3 things. You want to make sure that you attend the lecture, and once you have attended the lecture, you want to go in and take the quiz, online quiz. Okay, this is going to make sure that you get credit. Now, if you attend the live lecture, you're guaranteed 80%, a minimum of 80% in the participation grade. If you don't attend the live lecture, this is your way of making sure you get participation grade, points. Okay, take the online quiz. Now, the second thing you want to do is to do the assignment for the week. So every week there is an assignment, and it is due. The due date is already mentioned in the in the weekly course material, right? So you want to do the assignment. So for example, this week you were asked to pick a product, right? To pick a product and indication for that. So assignment, and the last thing you want to do, you want to make sure you do, is participate in the discussion forum. Okay, so as long as you're doing those 3 things each week, take the quiz, do the assignment, participate in the discussion forum, you will do very well in the class. Okay, and the assignments are, shouldn't take a lot of time, maybe half an hour at the most, right? Quiz also should not take a long time, as long as you have studied, you have attended the lecture or studied through, reviewed the lecture through the archives. It's an open book, open notes quiz, so you can have your lecture notes available with you when you take the quiz. And if you have studied the lecture, you should be able to do well in the quiz. Okay. And discussion forum, take a little time to do a little research on the questions that I have asked. And so, I would like to see a lively discussion. I think some people are really participating well. There are others that have not done so far, so I would like to encourage them to do that. So if you do that, you will be doing really well in the class. Okay, so any questions?
Professor Subhash, this is Victoria. I don't believe you're able to see the chat feature. No.
The chat feature?
Yes. Nobody's really asked a question, but just to let you know, I don't think you can see our responses.
Oh, yeah, that's right. Thank you. Thank you. Now I can see. I had to go through the okay, I get it. So sorry about that. I had to literally drag down the chat feature. I can see the chat feature, but I wasn't scrolling it down. Okay, so now I see Jerry saying, I don't think you can see the comments. Can you see the comments? Okay, thanks for telling me, Victoria. Next time I will make sure I will keep scrolling it down. So I see that you have answered 100 million for preclinical. Okay, so thanks for that. And sorry about that. I should have been scrolling it down. Okay, I get that. Thank you very much. Okay, any questions? I can see the comments now. So if you have questions, let me know. I see that you have said, I'm awake too, etc. Okay. All right. Jerry seems to be typing something. Okay, Jerry is asking, for other countries, do they have to follow this standard also? Yeah, so remember we talked about ICH, International Conference on Harmonization. So the countries that were party to that ICH, they expect the same standards. They expect the same standards to be followed. So more and more in different areas of clinical development, they are doing this now, the ICH thing. Okay, so they develop standards for laboratory studies and for clinical studies, etc., etc. So yeah. Now, of course, there may be some countries that don't have that, but most developed countries are following the ICH guidelines now. Okay, good question. Okay. So if there are no other questions, we'll stop the lecture here. And I want to again encourage everybody to participate fully. And I will see you online next week at this time. Jonathan seems to be typing something. Okay, Jonathan says he had a question about pharmaceutical product idea we had to submit recently. Okay, go ahead. Yeah, you had chosen the company GlaxoSmithKline, I remember, and you had chosen a small molecule product, right? So I encourage you to look up a protein product if possible. I think Glaxo has several protein products as well. And the reason is this is a biotech class, so I want to try to focus on. Okay, so a product called Twinrix, which is used to treat hepatitis. Yeah, this is most likely a protein product. Can you find a little more information about it and see if it is administered via injection or by orally? That will tell you. So try to get more information about it, but I think that might work. Okay. Yeah. So okay. So that seems to me that's a large molecule. Should be a biotech product. Okay. That sounds good. So Jerry is asking, when the FDA declines a product, can the company resubmit it, or is it completely shut down? Well, it really depends on what reasons they refuse permission. Sometimes they will say we need more data. Okay, so they will say we cannot give you permission right now because we need more data, and they will tell you that we need more data on this in this particular area. And so the company will go back and do more trials and provide the data, and then it will be approved. Sometimes the FDA will reject a product based on their inspection of the company, and they will say, you need to do more validation. So then you can again resubmit. But sometimes the FDA just says, we don't think this product is, they might say the product has insufficient efficacy and too many risks. So, and there are other similar products available that do the same thing. So they will reject it on that ground. In that case, it's shut down, and you don't go further. Does that make sense? So it's a case by case thing. Sometimes they will just tell you to do more studies. Okay. All right. So I will see you next week then. Okay. I'm going to stop recording now. You're welcome.
Thank you so much.
Bye, Osvaldo. You need to get on the online into the D2L. Okay. Let me know if you have trouble doing that.
I'm trying to do this from work, and that was killing me. I think the firewalls here do not allow me to test and everything, but